Hello, in this video we're going to look at enzyme inhibition and how enzyme inhibition affects the rate at which an enzyme controlled reaction can proceed. So we'll look at what an enzyme inhibitor is and the difference between a competitive and a non-competitive inhibitor, and then we will try to apply that understanding to some graphs. So firstly, and very generally, enzyme inhibitors are substances that will interfere with enzyme activity. And we are interested in those ones that slow down the rate of enzyme catalyzed reactions. Now, they can be competitive or non-competitive. And we'll look at this in a bit more detail, but broadly, competitive inhibitors are directed towards the active site of an enzyme, and a non-competitive inhibitor is directed towards a site away from the active site. So let's consider competitive inhibitors first. Here we have an enzyme. This is the substrate for that enzyme. So when looking at questions that relate to inhibition, it's important first to identify the enzyme, to identify the substrate, and if that question is about an inhibitor, to try to identify the inhibitor as well. So a really good tip is to identify each of these, the enzyme, the substrate, and the competitive inhibitor, before you proceed. Okay, so we can see the enzyme can either form a normal enzyme substrate complex. So here we've got that enzyme substrate complex that forms when the complementary substrate binds to that specific tertiary structure of the active site. If this were to happen, the reaction would proceed and a product would be formed. Again, it's helpful to identify the product when completing questions on this. Alternatively, a competitive inhibitor may have a similar shape and a similar charge. Now, similar is the key word here. You will lose credit for saying they are the same, because obviously they're not the same, but they have a similar shape and charge. And as they have a similar shape and charge, they can compete with, that's where that idea of competitive comes from, the substrate for the active site. So an inhibited enzyme has this competitive inhibitor bound to the active site that will block the active site of the enzyme and either stop or reduce the production of this product. Okay, so in most cases, a question about competitive inhibitors can be answered by saying that the inhibitor binds to or blocks the active site, and I'm emphasizing active site here rather than enzyme. So binds to or blocks the active site of the enzyme, which prevents or stops, slows down, reduces the production of whatever the product is. Okay, so if you've identified the enzyme, if you've identified the substrate and the competitive inhibitor, it's also useful to identify that product as well. Now we'll look at some examples of that. So the first one, and this is in the notes, so starting on page 23, but page 24 in the notes shows this, the inhibition of what's happening there? Shows the inhibition of this enzyme, succinic dehydrogenase. Now, in most cases, in fact, always, it would be enough just to refer to it as the enzyme. Okay, don't be confused by terms that you're not familiar with. They may be novel to you, they may be new, you may never have heard of them. But the important thing here is this is the enzyme. So we've identified the enzyme. We want to identify the substrate. It's done that for us here. So the substrate is succinate. That's a bit smaller, there it is a bit bigger. So succinate, and the product is fumarate. Now these are all substrates and products involved in respiration, in the Krebs cycle of respiration. So we've identified the substrate, we've identified the product, we've identified the enzyme. The last thing to identify is the competitive inhibitor, which in this case is malonate. Okay, now what can we see? The malonate, the inhibitor, and succinate, the substrate, have a similar structure. We can say they have a similar shape. And they've also got, if you notice down here, here and here, they've got a similar structure and a similar charge. And therefore, the competitive inhibitor, malonate, is able to bind to and block the, remember it's the active site that you must refer to, of the enzyme. So you could be specific in your response here and refer to it as succinic dehydrogenase, or you could just say the enzyme. That will be worth 1 mark. The second mark in a question on competitive inhibitors would be stopping the production or reducing the production. Stop or reduce production. Production of, well, we know our product would normally be fumarate. So in this case we can be specific. We can say it stops the production of fumarate, or it just stops the production of the product. Okay. Now, many enzymes will have more than one competitive inhibitor. Okay, so succinate dehydrogenase, which is the same as succinic dehydrogenase, has that, what did we say it was here? Malonate, which acts as a competitive inhibitor. But also, we can see that these other molecules, which have similar structure and similar charge, are also able to compete with the substrate for the active site. They bind to and block the active site, which prevents the formation or slows down the formation of the product, fumarate. Okay, so to understand graphs that might be related to this, here is an example. So this is a general trend that you would expect to see if we were to compare two enzyme-controlled reactions, where in the first reaction, there is no inhibitor present. In the second reaction, the competitive inhibitor has been added. Now you may notice that this no inhibitor present, so no competitive inhibitor present, it has the same same outcome, same shape of the graph as a normal substrate concentration graph. Okay, we can see on the x-axis we're increasing the substrate concentration, so that is the independent variable. On the y-axis, the dependent variable is the rate of the reaction. So as the substrate concentration increases initially, so does the rate of the reaction, until we reach this point here where there is no more increase in the rate of reaction, and that is because the enzyme active sites have all become saturated and they are working at their maximum rate, so the rate of production is greatest. If we compare that to the presence of a competitive inhibitor, okay, so if we add a competitive inhibitor to the reactions, we've now got the substrate plus the enzyme plus the competitive inhibitor in this reaction mixture, we can see that the rate of the reaction is lower at any given concentration of substrate. Now, the reason for that is because the competitive inhibitor is binding to and blocking the active sites of some of these enzymes, and therefore that prevents the formation of an enzyme substrate complex, which slows down the rate at which the product can be formed. You may also notice, though, that by increasing the substrate concentration to a really high substrate concentration, it is possible to achieve the same maximum rate of reaction. Okay, so competitive inhibition can be reduced with the addition of more substrate, as at high substrate concentrations, the substrate outcompetes the inhibitor. The substrate binds to the active site of the enzyme, and the products are formed after the enzyme substrate complexes have formed. So it's possible to say that the competitive inhibitors can be overcome by high concentrations of substrate. At lower concentrations of substrate, obviously, we can see that the rate is lower when the competitive inhibitor is present. So, one demonstration of this is in this is a tricky question that was on an exam paper a couple of years ago, relating to Alzheimer's. So as I said at the beginning, often these questions may be quite complicated and you may need to extract information from the stem of the question. So it's important to identify the enzyme, the substrate, the product, and any inhibitors. Okay, so if we read through this, Alzheimer's disease leads to dementia. It involves small beta amyloid proteins binding together to form structures called plaques on the brain. Okay, so Alzheimer's linked to these small beta amyloid proteins. Okay, those beta amyloid proteins, therefore we're going to consider the product. Okay, nerve cells in the brain produce large protein called amyloid precursor proteins. that has a complex shape. Okay, this protein is the substrate. Okay, so it's identified it for us. So those amyloid precursor proteins are the substrates of two different enzymes, alpha secretase and beta secretase. Okay, so we've not identified the enzyme just yet, but if we read on, these enzymes are normally produced in the brain. One product of the reaction catalyzed by the beta secretase is a smaller protein that can lead to beta amyloid formation. Okay, so we said earlier on that that beta amyloid protein was going to be our product in this case. So the beta secretase then must be the enzyme. We read on, many people with Alzheimer's disease have mutations that lead to alpha secretase production decrease alpha secretase production or increase beta secretase production. Now, this isn't actually going to be relevant for the question that we're going to answer in a second, but it might be interesting to see that this mutation, if you've got more beta secretase, there is a potential for producing more of this beta amyloid protein. Okay, one possible type of drug for treating Alzheimer's is a competitive inhibitor of beta secretase. So this is where this question that's going to follow, what we're looking at. Okay, when some of these types of drugs were trialled on patients, the trials had to be stopped because some patients developed serious side effects. Anyway, I'll ignore that bit for now, but we will consider this this idea of, well, we have competitive inhibitors for beta secretase. So we've got this competitive inhibitor. In the question that followed, one possible type of drug for treating Alzheimer's disease is a competitive inhibitor of beta secretase. Explain how this type of drug could prevent Alzheimer's disease becoming worse. Well, using that information that we came up earlier on, we can assume that the drug has a similar, sorry, I lost my pen for some reason, there it is. The drug must have a similar shape and charge. Having that similar shape and charge is what makes them competitive inhibitors. They must be able to bind to the active site. Remember, it's the active site that they bind to specifically, which would prevent the formation of the product. And we know that the product that causes that Alzheimer's, from reading through here, is those beta amyloid proteins. Okay, so you perhaps, if you saw competitive inhibitor and you'd read through all of this, you could have almost predicted what the question was that was going to come up. And this is why it's really important to try and extract information from these extended reading or sort of comprehension tasks type questions. Okay then, so in fact this question, it may be quite daunting the first time you read it, but for 2 marks it's quite straightforward. So we know the drug, the inhibitor, binds to the active site of the enzyme that we've identified already, which is beta secretase. There's the first mark. Okay, you could call it beta secretase, you'd get away with calling it just the enzyme. The second mark is it stops or reduces the production of the product. And we've identified the product already, those small beta amyloid proteins. For the second mark. Okay, so that was perhaps quite daunting for some students that completed that question. And in fact, if we have a look at the examiner's report for that question, oh sorry, wrong one. So 46% obtained a mark for the competitive inhibitor. Only 30% were able to go on and get that second mark. Okay, really that question, it's daunting because you've got all of this information, but if you break it down, if you identify the enzyme, the substrate, the products, and the competitive inhibitor, that should make it a bit easier. So that's competitive inhibition. Another type of inhibition, or the other type, is a non-competitive inhibitor. Okay, so non-competitive inhibitors usually involves a reversible binding of a molecule at a site away from the active site. So remember, in competitive inhibitors we always refer to binding to the active site. Here we always need to refer to the non-competitive binding away from the active site, or binding to a site away from the active site, or you may see it referred to as the allosteric site. Allosteric just describes a site away from the active site. Okay, the amount of inhibition, the degree of inhibition, is dependent on how much of this non-competitive inhibitor is present. And let's see the effect. Okay, so when a non-competitive inhibitor binds to the site away from the active site, it causes a change in the shape of the active site. Okay, so the tertiary structure of the active site is changed. That prevents the formation of an enzyme substrate complex. Okay, so the active site, it's changed shape, is no longer complementary to the substrate. The substrate is unable to bind, so there's no enzyme substrate complex, and therefore no product. Compare that to here, so without that inhibitor, without the non-competitive inhibitor, the active site remains in its complementary shape to the substrate, and a product could then form. Okay, so 2 marks for non-competitive inhibitor. First, binds away from the active site, and then the second changes the shape of the active site, preventing the formation, whoopsie daisy, what have I done there? Preventing the formation of an enzyme substrate complex. Okay, so if we look at the graph for this type of inhibition, you'll see that the no inhibitor present again is the same as the substrate concentration graph. Okay, by increasing the concentration of the substrate, it will increase the rate at which the reaction can proceed. Okay, just as we mentioned earlier on, obviously at this point the Vmax, that is when all of the active sites are occupied, they're saturated, and therefore it's the maximum rate at which an enzyme can combine with a substrate, or the maximum rate at which the enzyme can form enzyme-substrate complexes. And that's happening at this Vmax. Okay. At this point, all of the active sites are saturated with substrate. If we compare that to the addition of a non-competitive inhibitor, we can see that it reduces the Vmax. So unlike the competitive inhibitor, increasing the substrate concentration to a high substrate concentration will not overcome the effect of the non-competitive inhibitor. That same rate of reaction, that same maximum rate, cannot be achieved when the non-competitive inhibitor is present. Okay, so non-competitive inhibitors cannot be overcome by increasing the substrate concentration. Let's have a look at an example of this then. So, firstly, let's read through the stem. Let's see if we can identify the enzyme, the substrate, and the products. So the rate at which phenol was broken down by the enzyme phenyl oxidase. So phenol must be our substrate. By the enzyme phenyl oxidase was measured in solutions with different concentrations of phenol. The experiment was repeated with a non-competitive inhibitor added to those phenol solution. The graph shows the results. So fairly typically, in a question like this, you may have a comparison between the reaction without the inhibitor and with the inhibitor. So another fairly typical question would be to explain the difference in those two curves, and in this example, explain why an increase in concentration of phenol solution from 2 to 2.5 millimoles per decimeter cubed has no effect on the rate of the reaction without the inhibitor. So we're looking at without the inhibitor from 2 to 2.5 millimoles, so there is from there to 2.5 there, so increasing the concentration of the substrate but no effect on the rate of the reaction. And we've mentioned this already. At this stage, it is the maximum rate at which the enzyme can form enzyme substrate complexes. Okay, that would be the first mark. The second mark is just that the active sites are saturated with substrate. Okay, 2 marks there. Essentially, this is a question about increasing the substrate concentration. But it's fairly typical that you'll see that combined with an inhibitor question. Okay, in the second part of this question, explain the effect of the non-competitive inhibitor. So, here is our with the non-competitive inhibitor, we're explaining the effect. So we don't need to discuss the shape of the curve. We don't need to describe it, we just need to explain why. So, a non-competitive inhibitor binds to the enzyme at a site away from the active site. Okay, for the first mark, we say what the non-competitive inhibitor does. It binds to the enzyme at a site away from the active site. For the second mark, we know that the non-competitive inhibitor changes the shape of the active site. So the active site is no longer complementary to the substrate, so that prevents the enzyme substrate complexes from forming. Okay, no enzyme substrate complexes, that reduces the rate at which the product is formed, or reduces the rate of the reaction. Excellent. So, by the way, I've used this ES complex. It's safer for you to write enzyme substrate complex. Perhaps I'm rushing a little bit. Okay, so there are two fairly typical questions on competitive inhibitors and non-competitive inhibitors. In summary, we can use this slide. You may wish to add annotations from this slide to your notes, but we have covered each of these points for the two different types of inhibitor. Okay, I'll leave it there. Thanks for listening along.